Then pacemaker enzyme. This is a question for pre-medical. This enzyme is phosphofructose kinase. This is the pacemaker of enzyme. Why pacemaker? Glucose converted into glucose 6-phosphate. This is hexokinase enzyme. This is hexokinase. Then glucose 6-phosphate converted into fructose 6-phosphate by isomerization. Then fructose 1,6-diphosphate. It uses the ATP again. Here the use of ATP, here also use of ATP. What is formed? Fructose 1,6-diphosphate. And this is the process of charging completed, charging of substrate. From this, glycolysis proceeds. So this enzyme is phosphofructose kinase. Phosphofructose kinase. It is termed as pacemaker enzyme. Pacemaker enzyme. And this enzyme is marker enzyme of mitochondria. Last one is marker enzyme. These two questions are useful. Marker enzyme of mitochondria succinate dehydrogenase. Marker enzyme of mitochondria succinate dehydrogenase. Marker enzyme of mitochondria succinate dehydrogenase. Question number 14. Aldolase. Aldolase enzyme is responsible for the condensation as well as cleavage. Condensation means combining, and cleavage, that is splitting. So fructose 1,6-diphosphate. Fructose 1,6-diphosphate. Short form. F for fructose, 1,6 position of phosphate group. With the help of enzyme aldolase. With the help of enzyme aldolase. With the help of enzyme aldolase, converted into 3C compound, 3-phosphoglyceraldehyde and dihydroxyacetone phosphate. DHAP, that is dihydroxyacetone phosphate. See that glycolysis again. See this one. See this table. This is the use of aldolase enzyme. It splits fructose 1,6-diphosphate, that is by the process of cleavage. By the process of cleavage, converted into these two. This is the activity of aldolase. And one more speciality, if required, then these two can combine and back to form what is fructose 1,6-diphosphate. So this is the multiple utility of this cycle actually. You make the cycle and learn properly, and you are able to attempt number of questions related with the glycolysis. So finally, the answer for the 14th question, that is splitting of or cleavage of fructose 1,6-diphosphate into DHAP 3C compound, PGAL 3C compound, dihydroxyacetone phosphate. And what is PGL? Phosphoglyceraldehyde. AL stands for aldehyde. This is the use of enzyme aldolase, responsible for condensation as well as cleavage. This enzyme responsible for cleavage or splitting, and if required then condensation. Both activities is done. That is work in both directions depending upon the requirement of cell. That is cleavage as well as condensation. One molecule of sucrose and pathway is EMP. Actually glucose splits into 2-pyruvic acid. 2-pyruvic acid. It's a 3C compound from one glucose. So if we take sucrose, sucrose is C12 H22O11. First of all, by the addition of water, get converted into C6H12O6 plus C6H12O6. That is, two glucose molecules are there. So from one glucose, 2 pyruvic acid. From one glucose, from one glucose, 2 pyruvic acids are there. Then from 2 glucose, how many pyruvic acid? That is 4 pyruvic acid. 4 pyruvic acids are formed. Sucrose splits into 2 glucose and each glucose provides 2 pyruvic acid. Sucrose is converted into what? That is 2 molecules of glucose and each one give 2 pyruvic acid. So total number of pyruvic acid or pyruvate, that is 4 from one sucrose molecule. This is Krebs cycle, actually second part. Number of questions are also based on this Krebs cycle. That is after glycolysis. This is the glycolysis. Its product is pyruvic acid. This is end product of glycolysis. But before entering in Krebs cycle, get converted into acetyl coenzyme. It's a 2C compound. It is intermediate step. This is link step. Actually. Link reaction. This is link reaction. It is performed in between glycolysis and Krebs cycle. And you see, there is a oxidation as well as decarboxylation. Oxidation and decarboxylation both takes place in link reaction. So this step results in the formation of acetyl coenzyme and this is first decarboxylation. This step is known as oxidative decarboxylation, because oxidation as well as decarboxylation both are involved. Oxidative carboxylation is seen. Why oxidation? See this one. This is oxidation and this is decarboxylation. So collectively termed as what? That is oxidative decarboxylation. Then this acetyl coenzyme oxidizes completely by Krebs cycle. This acetyl coenzyme oxidizes by the Krebs cycle and final product of Krebs cycle is H2O plus CO2. That is complete oxidation of acetyl coenzyme takes place by Krebs cycle. So acetyl coenzyme A is connecting link. Acetyl coenzyme A is connecting link between glycolysis and Krebs cycle. Then see, these are the different steps. These are the different steps of Krebs cycle. That is citric acid, succinic acid, isocitric acid, oxalosuccinic acid, alpha ketoglutarate acid. You can this is very easy to learn. That is, take the initial letter of each acid. C is OK. These are the first 5 acids. Citric acid, succinic acid is for isocitric acid, O for oxalosuccinic acid, K for ketoglutaric acid. The first 5 steps, that is C is OK. C is OK, first 5 steps. Second 5 steps, S, few, moxa. moxa, and sentence framed as yes, few moxa. Yes, few moxa. Then S for succinyl coenzyme है, succinic acid, fumaric acid, malic acid, oxaloacetic acid, oxaloacetic acid. So these are the 10 different acids. First of all, you learn their sequence. That is C is okay. Yes, few moxa. Then how did they form? That is already mentioned in the Krebs cycle. This is already mentioned in the Krebs cycle. So number of questions are based over this process. Let's see. First, connecting link between glycolysis and Krebs cycle. Connecting link between glycolysis and Krebs cycle. See the answer. That is acetyl coenzyme A. I have already mentioned this is acetyl coenzyme A, connecting link between glycolysis and Krebs cycle. Then second one, Krebs cycle starts with, see this cycle again. This is 4C compound, oxaloacetic acid, and combines with the acetyl coenzyme A. See this one. Oxaloacetic acid combines with the acetyl coenzyme A to form citric acid. That is 4C plus 2C, formation of 6C again, that is citric acid. So see the question. Condensation of 2C, that is acetyl coenzyme A with oxaloacetic acid. So we can write it as, that is acetyl coenzyme A, 2C compound combines with 4C oxaloacetic acid. This is the way by which you can easily know. So that's why this cycle is important to attempt such type of questions. So Krebs cycle starts with the condensation of 2C compound with the 4C compound. 2C compound is acetyl coenzyme A, oxaloacetic acid is 4C compound. And citric acid is formed. Its result is citric acid. So in Krebs cycle, citric acid formed as initial product and get oxidized again into the oxaloacetic acid by different pathway. So Krebs cycle is also known as citric acid cycle. See this one. Citric acid is formed and get oxidized again into oxaloacetic acid. Oxaloacetic acid plus acetyl coenzyme A collectively forms citric acid. Then it oxidizes into various paths, various reaction, and back to form what? Oxaloacetic acid. So that's why Krebs cycle is also called citric acid cycle. Krebs cycle is, its other name is citric acid cycle. And citric acid contains 3 COOH group. COOH group. These are 3. Their number is 3. So that's why citric acid is tricarboxylic acid. Citric acid is tricarboxylic acid. Hence it is also called citric acid cycle. Krebs cycle is also called citric acid cycle. Which of the following enzyme of mitochondria? Which of the following enzyme of mitochondrial membrane? Yes, Krebs cycle. Most of the enzymes, back to the Krebs cycle again. That is, these are the various types of enzymes participates in the Krebs cycle you see. Number of enzymes are associated with the each step. And most of the enzymes are found in the matrix of mitochondria except this one. This enzyme is found in mitochondrial membrane. This is inner membrane enzyme, succinate dehydrogenase enzyme. This is from the inner membrane enzyme. Succinate dehydrogenase enzyme are found in inner membrane. It is a part of electron transport system and it is marker enzyme of mitochondria also. Succinate dehydrogenase enzyme, number 1, it is a marker enzyme of mitochondria. Marker enzyme of mitochondria. Marker enzyme of mitochondria. And it is located in inner membrane. It is located in inner membrane. So we say membrane bond enzyme. This is membrane bond enzyme. It is a part of electron transport system also. That is complex 2. Enzyme of complex 2. Enzyme of complex 2. So 3 possible questions from this enzyme. Rest of the enzyme, these all enzymes are found in matrix of mitochondria. These are found in the matrix of mitochondria. Citrate synthetase, malate dehydrogenase and enolase enzyme. They are found in, first 2 enzymes are found in mitochondria, these 2. And this is in the cytoplasm. This enzyme is found in cytoplasm and involved in the glycolysis. Enolase enzyme is found in cytoplasm and involved in glycolysis. In which process? That is dehydration. TCA, tricarboxylic acid. Substrate phosphorylation takes place. Substrate phosphorylation means that is removal of phosphate. See this one, succinyl coenzyme A. Then back to refer that cycle, please wait. That is succinyl coenzyme A. It is 4C compound. When it get converted into succinic acid, it is converted into succinic acid or succinate. There is a loss of phosphate. There is a loss of phosphate group. PO4 is released. That is decarboxylation of succinyl coenzyme A. But this phosphate is immediately trapped by guanosine diphosphate. GDP stands for guanosine diphosphate. It is converted into GTP, guanosine triphosphate. That is transfer of phosphate group at substrate level. This substrate releases phosphate and this substrate of this reaction receives phosphate. So there is a transfer of phosphate group. There is a transfer of phosphate group at substrate level. Hence it is called substrate phosphorylation. That's what is known as substrate phosphorylation. So please back to the Krebs cycle again. See this one. Succinyl coenzyme A is converted into succinic acid. Then you see there is a loss of there. This is triphosphate, this is diphosphate. Definitely phosphate group is released from this one and taken up by the guanosine diphosphate. So this is the only step where substrate phosphorylation is observed in Krebs cycle. And the product is different. In glycolysis, it was ATP. In glycolysis, it was ATP, whereas in Krebs cycle, it is GTP. It is a GTP, that is guanosine triphosphate. So substrate phosphorylation takes place in between succinyl coenzyme A to succinic acid. This path, again and again, be better to learn this activity, learn this process line to line. You will be able to attempt number of questions related with the Krebs cycle. So come to that question, that is this one. So my answer, that is succinyl coenzyme A 4C compound. It undergoes dephosphorylation, that is release of phosphate by succinyl coenzyme A. Release of phosphate by succinyl coenzyme, it is 4C compound and taken up by the guanosine diphosphate, then only the GTP formation is possible. So this is indirect question, that is you must learn each and every step of Krebs cycle and use your full knowledge for attempting such type of questions. The only C5 compound, see that the simple one, C is okay, yes, few, moxa. These are the 10 acids of Krebs cycle. And alpha ketoglutaric acid is C5 compound. Prior to this, all are C6, and after this, all are C4. After that, all are C4. This is the way by which it can easily be done. And if you want to check it, then back to the Krebs cycle again. See this one. This is 6C, 6C, 6C. Then it becomes 5C for the first time. Becomes 5C for the first time. And alpha ketoglutaric acid is formed. That is, before this all are 6C. After that up to oxaloacetic acid all are C4. Because again alpha ketoglutarate glutarate loses carbon dioxide. So all these acids after ketoglutaric acid are 4C. Before ketoglutaric acid they are 6C. So there are two places from which carbon dioxide is released. Carbon dioxide is released the space that at this step number 1. If carbon dioxide is released, that's why C5 becomes C4, C6 is converted into C5. And if ketoglutaric acid releases CO2, then only 4C compound is possible. So it also indicates in which steps carbon dioxide is released. In which step carbon dioxide is released. And one more thing, this oxaloacetic acid forms what? Citric acid by combining acetyl coenzyme A. By combining acetyl coenzyme A. So answer for this question, that is alpha ketoglutaric acid is the only 5 carbon compound formed in Krebs cycle. Then TCA cycle number of GTP I have already mentioned, that is step from 1 Krebs cycle 1. From 1 Krebs cycle 1, because here see this number 1 acetyl coenzyme A is given. If 2 acetyl coenzyme A are there, then we can say 2. Actually see this one. In glycolysis, glucose forms 2 pyruvic acid. Glucose forms 2 pyruvic acid. Then each pyruvic acid converted into acetyl coenzyme A, 2C compound. Similarly, this pyruvic acid is also converted into acetyl coenzyme A, acetyl coenzyme A. That is after glycolysis, every step takes place twice. And this acetyl coenzyme A performs 1 Krebs cycle, and this acetyl coenzyme A performs 1 Krebs cycle. Krebs cycle, that is complete oxidation of acetyl coenzyme A. So after glycolysis, this is the glycolysis end product. This is intermediate step, double and double Krebs cycle. Double Krebs cycle, double intermediate steps. But here question is already given, that is 1 acetyl coenzyme A, that is 1 Krebs cycle is sufficient, and the answer is that is 1 GTP from 1 Krebs cycle.